Thank you. Thank you, the organizers, for letting me visit with you all. This is a pedigree that we saw in the, I guess, early 80s. These people had retinitis pigmentosa with spicular formation, and most people would have called that an autosomal dominant disease, except for this woman came to our clinic, and she was holding this child in her arms, and this child was bumping into the walls as he walked down the corridor. And so that did not look very much like retinal degeneration. It turned out that on talking about this pedigree, this gentleman had reduction in the cerebellum and degeneration of the brainstem, olivopontocerebellar atrophy, and he was in a rest home. And so then we began to realize that these people were all related in a very unique way, and that is through the mitochondrial DNA or the maternal inheritance. And they have a mutation in a part of the mitochondrial energy generating system called the ATP synthase, and it changes this leucine to a glutamate, and the glutamate then blocks the spinning of this wheel that's necessary to generate the ATP. And these people have a mutation in this particular mitochondrial DNA gene, and the percentage of the mutant mitochondrial DNA determined how severe their phenotype was. So let's see. All right, so what are we talking about here? We're talking about this piece of DNA that's in the mitochondria, the power plant of the cell, and this codes for critical genes that are involved in energy metabolism. The mitochondria is also composed of polypeptides that are also located in the nucleus. There are about 1,000 nuclear-encoded proteins and 13 critical polypeptides that are necessary for making energy by the process of oxidative phosphorylation, complex 1 to CoQ to complex 3 to cytochrome C to complex 4, and then to ATP synthase. And basically, what you're burning is hydrogens in your food. They're carried through NADH to the electron transport chain. The electrons flow down the electron transport chain. The energy is used to pump protons across the inner membrane to create a capacitor, and that capacitor is used by potential energy as potential energy by the ATP synthase to make ATP to do work. So you're coupling oxidation with phosphorylation, hence oxidative phosphorylation. And this system generates much of the reactive oxygen species that Josh was just talking about, and there are systems to detoxify them, manganese SOD to take superoxide and hydrogen peroxide, and glutathione peroxidases to take hydrogen peroxide and oxidize materials to less toxic components. The mitochondria also regulates calcium, and it has a self-destruct system, the permeability transition pore, that causes or drives apoptosis. So this shows you the mitochondrial DNA and the mitochondrion, and the mitochondria is involved in energy metabolism, redox balance, generates reactive oxygen species, regulates calcium, regulates apoptosis, and is critical in regulating the nucleus through the epigenome. So basically, the mitochondrial DNA is a circular molecule of 16,259 base pairs, codes for a regulatory region, a small and a large ribosomal RNA, 13 polypeptides, 7 for complex 1, ND 1, 2, 3, 4, L, 4, 5, and 6, 1 for cytochrome B, complex 3, and 3 for complex 4, CO1, 2, and 3, and 2 for the ATP synthase, complex 5. The mitochondrial DNA is inherited exclusively from the mother, goes from mother to all of her children, and daughters to their children, but male's mitochondrial DNA are destroyed and do not contribute. And that is because you do not want the mitochondrial DNA to recombine, because different mitochondrial DNA wiring diagrams of the power plants have different energetic efficiencies, and if you mix the components, you would short the circuit, and that would be lethal. So, in fact, there is no recombination. The cell has thousands of copies of the mitochondrial DNA, and they're constantly mutating inside our cells, and that creates a mixture of mutant and normal, and we call that heteroplasmy. And as the cell undergoes division, these can segregate, give you different percentages of mutant mitochondrial DNA, and that as the number of mutant mitochondrial DNAs increases, the energy declines, and when that goes below the minimum for that organ or tissue to function, then you get a phenotype, and the retina is the most oxidative organ in the body. The mitochondrial DNA has accumulated mutations as women have migrated out of Africa and around the world, and every time women moved into a new area, they acquired mitochondrial mutations that adjusted their energy metabolism and allowed them to adapt to different environments. And as we age, we accumulate somatic mitochondrial DNA mutations, and if we put in an antioxidant defense into the mitochondria, we can actually reduce the mitochondrial DNA damage and increase the lifespan. And I would suspect that some of the damage that Josh is seeing is in the mitochondrial DNA. So this is a pedigree where these individuals had optic atrophy. They're related to the maternal lineage, to this woman, and to all of these people. These individuals, this individual had optic atrophy, as did these individuals. This individual had optic atrophy. She had two different consorts, and each of these had dystonia. So this is a person trying to get out of her wheelchair, and she's rigid with bilateral basal ganglion degeneration. This is due to a mutation in the mitochondrial DNA that is heteroplasmic, and those people that have dystonia have pure mutant, homoplasmic. Those people that have optic atrophy are heteroplasmic, so a quantitative energetic deficiency. So this is changing this very highly conserved amino acid to valine, and there's a milder mitochondrial mutation in the same protein, ND6, which gives optic atrophy. This one gives you optic atrophy and dystonia because it's more severe. Now, this is another pedigree with a different mitochondrial DNA mutation. In this case, this is in the ND4 gene at 11778. These people all have central vision loss. That's a late onset loss of central vision in one eye and then in the other, called Leber's hereditary optic neuropathy. It's a male-biased disease, and that's because estrogen is protective of mitochondrial dysfunction, and so you bias male expression. Now, there are multiple different mitochondrial DNA mutations that cause Leber's hereditary optic neuropathy of different severities, and the mildest mutations have to have a second event to allow penetrance of the disease, and that can either be due to changes in this mitochondrial lineage, the background that we were talking about, or other mutations such as this mutation in ND1. So why are we getting optic atrophy when we have a systemic mitochondrial defect? Well, that's because, according to Alfredo Sadun, this portion of the optic nerve, which is across the retina, has to be clear, and therefore it cannot be myelinated, and it takes a lot more energy to maintain the action potential in these region of the optic nerve, and so these fibers selectively die, and that gives you the central scotoma. We've then put these mitochondrial DNA mutations into cell lines with the same nucleus, and what you can see is they consistently show a defect in mitochondrial energy production and an increase in oxidative stress and an increase in apoptosis. So that's basically killing the optic nerve cells. And what's interesting is that as you increase the level of mutant mitochondrial DNAs, you increase the penetrance of the defect, and other factors such as smoking, which inhibits mitochondrial function, or alcohol consumption can increase the probability of onset of the phenotype. Mitochondrial haplogroups, lineages that I've mentioned, are also associated with glaucoma, and somatic mitochondrial DNA mutations are also seen in glaucoma. And in glaucoma, we see a decrease in respiratory complex 1, as we see in Leber's hereditary optic neuropathy, and the trabecular network has a high level of oxidative stress, which we think is important in the pathophysiology of that disease. We've also studied the mitochondrial DNA lineages and age-related macular degeneration. In this particular lineage, if you get this, you have a 100% probability of getting AMD. This is a pedigree that has a mutation in the ND6 gene at proline 25 deleucine. This individual had 50% heteroplasmy, it had optic atrophy and cerebellar ataxia. This individual had 5% mutant and was perfectly normal in her blood, but she had 3 consorts and every one of her children was 100% mutant and died of Leigh syndrome. This changes a very highly conserved proline to a leucine, and so we made a mouse in which we created a cell line where we mutated that particular gene to give you the proline to leucine change. We enucleated the cell, we made a female pluripotent stem cell, we fused in the mutant mitochondrial DNAs, we put it in a blastocyst, and we then introduced this mutation into female germline. So I'm going to talk about both an ND5 and an ND6 mutation. And this defect in complex 1 will then increase NADH because you're not oxidizing it. This is a fluorescence lifetime imaging showing the hippocampus has very high levels of NADH, and that increases the level of reactive oxygen species, resulting in optic fiber degeneration. These two mice have ERG alterations, but what's interesting is that they have different severities of complex 1 defect. This is a control, this is the ND5, and this is the ND6, only about 20% difference. But the difference is a marked difference in behavior. These are the different color animals, and this is a study of stress, and you can see that the wild type animal is less stressed than the ND5 and then the ND6. And if we look at depression, we can see the reciprocal. And then if we give them lithium, we can replace that defect. The ND6 animal has a defect in social interaction, so it's much more likely to interact with an innate object than a different mouse. It has obsessive compulsive behavior, and it has alterations in its EEG. This animal then basically has autism. So we can then use a variety of different approaches to intervene in mitochondrial energy metabolism. We can use different derivatives of ubiquinone. We can use cyclosporin A that blocks the permeability transition pore, but we can also take advantage of the fact that estrogen is protective, and if you put estradiol into the culture cells of labors, you can in fact recover much of the complex 1 defect. So basically then we have a new approach to looking at how we can get retinal degeneration disease and optic atrophy, and this may be an important component for a wide variety of retinal problems because of the very high oxidative demand of the retina. Thank you very much, and these are the folks that did the work.
Thank you. Questions?
Doug, what's the mechanism of the very interesting estrogen protective effect?
Thank you. So the question is, what's the mechanism of the estrogen protective effect? What we discovered a number of years ago, where is that? Well, anyway, sorry. What we discovered a number of years ago is that, oh yeah, here it is, that if we fractionate the cell and we look for the estrogen receptors, there's 2 of them, alpha and beta, it turns out that the estrogen beta receptor is strongly incorporated into the mitochondria. And if we then put estradiol on these cells, we can actually increase the manganese SOD antioxidant defense, doubling it within a matter of about 2 hours. And this, we believe, is an important factor for why there's a very strong male bias in a number of mitochondrial diseases, because women have routinely then upregulating their mitochondrial antioxidant defenses as they go through the estrous cycle.
So we see that's fascinating. So we've seen a sex bias in a lot of mouse models of retinal disease, and we've been doing some experiments with adding estrogen and seeing some very intriguing protective effects and wondering what the mechanism might be. And I think you might have just told us what it is.
Yeah. Well, we showed that a number of years ago, and we feel that's critical in labors, autism, Parkinson's disease, and maybe a variety of other ophthalmological diseases.
So does the estrogen receptor beta translocate to the mitochondria after it's activated by estrogen, or is it already on the mitochondrial membrane?
We don't know that for sure, but these are untreated animals right now, and you can see that there's still a lot of beta in the mitochondria. So I think it's a significant portion is targeted to the mitochondria to begin with.
Do you think this might be part of the reason that women, in addition to being superior to men, live longer?
Very much so. So we feel that the aging clock is the accumulation of somatic mitochondrial DNA mutations, and that diminishes your energy potential over time, giving you the age-related phenotypes of the oxidative organs, the brain, the heart, the muscle, and the renal, and that this estrogen protection then gives the extra 10 years that women have of the lifespan, we believe.
Thank you.
Other questions? Yes.
Are there any clinical trials of estradiol in labor patients to protect the second eye?
That's a great question, and the answer is no. Right now, most of the clinical trials have been looking toward regulating the apoptotic component and thus blocking the actual death of the retinal ganglia cells, but increasing non-feminizing phytoestrogens has been something actively being discussed. And there are some real good examples in traditional Chinese medicine that might work very well.
With anti-testosterone compounds, like prostatic carcinoma patients, is that in some way protective? Is it the balance or just the estradiol levels alone?
We don't think that the testosterone is, in a sense, toxic. Rather, the estrogen is upregulating the antioxidant defenses and is protective. So the converse would intervention would not be relevant.
Any other questions? Yes, go ahead.
Could you please comment a little bit on how the mutations or dysfunctions of mitochondrial DNA affects the genome or epigenome in the nuclei?
Yes, so the question is, does the mitochondrial DNA variation have some effect on the nuclear gene expression? And the answer is absolutely. So mitochondrial DNA mutations then are changing oxidative phosphorylation. That changes the oxidation of NADH to NAD. NADH is critical, NAD is critical for the TCA cycle because TCA cycle is oxidizing substrates and making NADH to be burned in the electron transport chain. So if you have an excess of NADH, you stall the TCA cycle. The TCA cycle then makes a large number of substrates, and those substrates are the substrates for the nuclear encoded epigenomic modification enzymes. So the mitochondria makes S-adenosylmethionine, that's for methylation. It makes alpha-ketoglutarate, that's for the Diamond-G demethylases. It makes a citrate that makes acetyl-CoA, and that's the regulation for acetylation of the nucleus. So the whole regulation of this nuclear cytoplasmic balance is critical. That's done through these small molecules and ROS.